and the Initiative for Food and Agricultural Transformation at the Ohio State University is to examine the soil, plant, water, animal, human nexus to enhance climate resilience of agroecosystems through adaptation and mitigation strategies that improve resource use efficiency, sustain productivity, restore soil ecosystem services, and improve carbon sink capacity. Prior to joining Ohio State University, Dr. Sprunger completed a National Science Foundation postdoctoral fellowship in biology at Columbia University, where she examined the relationship between soil carbon dynamics and crop productivity in smallholder farming systems in Kenya and Tanzania. Dr. Sprunger holds a PhD in crop and soil science and ecology, evolutionary biology, and behavior from Michigan State University. And she also received both her Bachelor's in Science and Forest Resources and Bachelor's in Art in Program on Environment from the University of Washington with a minor in Human Rights. So please help me welcome Dr. Christine Sprunger.
Thank you so much for that introduction. Can everyone hear me in the back? Okay, great. I'm not used to having this microphone, so please let me know if you can't hear me. But thank you so much for having me here today. This is actually my first time in Iowa, so always love to check off a new state. And I'm happy to be here today. So the question that I'm going to pose in this seminar is really this kind of cheeky title of understanding perenniality and diversity, and asking, which is most effective at providing ecosystem services within agroecosystems. So typically when I give seminars, I tend to talk to more ecology departments, and they aren't really aware of how prominent cropland is across the United States. But you all know that it is very prominent, and you, being the state of Iowa, are right at the center of this. While this cropland is, productive in terms of land acreage, very important for contributing to the U.S. economy, we know that this level of intensity has some downsides when it comes to the environment. And of course, we are well aware of environmental disservices that are caused by agriculture. Now that I'm at The Ohio State University, we focus and think a lot about water quality issues within Lake Erie that we've now come to realize are the result of phosphorus and nitrogen runoff from agricultural landscapes. We also know that through the intensification of agriculture, we have seen large areas of soil erosion across the globe. And it's important to also think about how we have these negative feedback loops. So of course, when we have soil disturbance like erosion, we're emitting carbon dioxide back into the atmosphere, creating global climate change. And it's important to think about how climate change itself is changing agriculture. So something that I think a lot about in Ohio is extreme weather events, and especially flooding. And so thinking about what are some ways in which we can combat these very serious environmental disservices. So the good news is there are several ways in which agriculture can actually be a part of the solution to actually provide ecosystem services and hopefully lead to mitigations of climate change, greater retention of our nutrients in our landscape. So I think a lot about recovering soil carbon, and we know that, especially in the Midwest, we have a large potential to recover a lot of the carbon that was lost. So this map here shows the amount of soil organic carbon that has been lost since pre-settlement, and the darker the color, the greater the amount of carbon that has been lost over time. And guess which state is the darkest? It's actually Ohio. But again, we do know that there are ways in which we can try to increase carbon in agroecosystems. We also know that mitigating climate change isn't the only benefit from this. We know that an increase in carbon can be related to improvements in soil health and crop productivity. the big question is which management practices are actually most effective at doing this. So some of the soil health promoting practices that I've focused on over the course of my career are thinking about how increases in organic matter inputs can influence soil carbon, thinking about the reduction in soil disturbance, so thinking about incorporating more no-till systems or reduced tillage systems in our landscapes, and then also thinking about year-round ground cover. So we've had a lot of discussions today about the incorporation of cover crops, and we know that there's still low adoption across the United States, but this is an effective way in which we can reduce disturbance and add organic matter inputs. We also know that just increasing diversity in agriculture is really important, and that can typically be done through the use of expanding our crop rotation. So actually a meta-analysis done by Dr. McDaniel, a member of your department. And what we see here is that an increase in crops in the rotation leads to an increase in total carbon and total nitrogen. So this shows that typically, when you get to numbers 4 or 5 plus numbers of crops in your rotation, it's clear that there's cover crops in the rotation that are really important for these nutrient pools. Another way that we can actually diversify our landscapes is thinking about growing perennial crops in the place of annual cropping systems. And the reason why we might want to do this is because perennials tend to have significantly greater root systems compared to their annual crops. So for example, this is a perennial intermediate wheatgrass that has been developed at the Land Institute in Kansas. And you can see that throughout the course of the growing season, or throughout the year, they have these prolific deep root systems, while the annual system only has a substantial amount of roots at peak biomass. So again, having a perennial in the system just leads to the opportunity to have living roots throughout the year that are constantly contributing to soil organic matter accumulation. So that was actually a big question in my dissertation. It was the first chapter where I really wanted to understand how does perenniality impact rhizosphere dynamics and contribute to ecosystem services. So again, we're comparing a perennial intermediate wheatgrass to annual winter wheat. We had hypothesized that yes, perennials will have greater root biomass, but we weren't sure how that related throughout the soil profile, and especially we weren't sure if this would stand if we actually managed perennial crops like we do our annual wheat systems with intensive fertilizer. So some of the questions that we asked were, do perennial crops allocate more biomass below ground compared to annual crops? And which crop is most effective at reducing nitrate leaching and enhancing nitrogen use efficiency? And then I also wanted to ask, are soil carbon pools greater under perennial systems due to larger roots? So over the course of 4 years, we basically compared annual and perennial systems and looked at each plant component to see how each plant was allocating energy. And so what we see here, in 2012, I hadn't arrived yet, so we actually weren't able to measure the roots, but 2011 was my first year, and we see that the perennial system allocates much more energy below ground, which is what we hypothesized. Interestingly enough, we do see that the perennials are still allocating quite a lot above ground, but where we see a decrease is in the seed yields. So that is one downside of why a farmer might not want to grow this perennial intermediate wheatgrass, because it yields so much lower than the annual wheat system. However, in thinking about ecosystem services such as carbon storage and the potential for reductions in nitrate leaching, we were very excited to see such large allocations below ground. So what did this mean for carbon? I looked at particulate organic matter size fractionations in the final year of the experiment, and sadly didn't detect any differences at the surface or in subsurface soils between the intermediate wheatgrass and the annual wheat. And this was really surprising to me because we saw that the perennial intermediate wheatgrass was consistently sending large roots down the soil profile. And so what could be the cause of why we didn't see differences in either the active or slow carbon pool? And I'll leave that as a little bit of a cliffhanger, but it was something that puzzled me in my first chapter, and I continued to think about the role that annuals and perennials can play in soil carbon accumulation. So we'll come back to this story in a little bit. In terms of the perennials' ability to reduce nitrate leaching, we actually saw that the annuals were leaching nitrates at an extremely high rate compared to the perennial crop, which shows that perennials are really effective at being able to retain nitrogen. So from a water quality standpoint, this is a really exciting finding that perennials are able to substantially reduce nitrate leaching. But one of the questions that I wanted to ask was, why are perennials able to reduce nitrate leaching relative to annuals? So I took this as an opportunity to do a simple nitrogen use efficiency balance and really just ask how much added N is actually used by each crop. And so we just used a mass balance approach to determine nitrogen use efficiency, where we looked at total crop nitrogen per total nitrogen fertilizer added. And we really wanted to assess both above and below ground nitrogen use efficiency to see, was this really the result of root systems? So when we looked at just the above ground nitrogen use efficiency, we actually don't see any difference between the annual wheat and the perennial intermediate wheatgrass, which is to say that from an above ground standpoint, they're using nitrogen the same way. However, when we look at the root system, we see extreme differences between the nitrogen use efficiency of the perennial and the annual system. And so then when we look at the whole plant nitrogen use efficiency, again, we see the same trend. So we're very comfortable in saying that, yes, it's the roots that are contributing to the retention of nitrogen. And one interesting thing to point out here is that any number where the ratio is greater than 1, that actually shows that the perennials are actually taking up more nitrogen than was actually applied. So it just shows that they're very effective at mining nitrogen and maybe too good at it. But again, just could be a useful tool and a way to retain nitrogen in our systems. So just a quick summary on this point. Perennials had at least 3 times more root biomass compared to annual wheat. Annuals leached nearly 5 times more nitrate relative to perennials. The perennials consistently had greater nitrogen use efficiency, and we can now say, yes, that was largely due to their root systems, but we did not see any differences in soil carbon pools after 4 years. So this last point really just was kind of something that kept nagging at me, and I was like, is this something that we'll see in all types of perennial systems? And so luckily, being at the Kellogg Biological Station at Michigan State University, we have this really nice biofuel cropping system experiment that has a gradient of annuals versus perennials, but also thinking about questions pertaining to diversity. So we go from continuous corn to corn in rotation at every entry point, And then we have our monoculture perennials, switchgrass, miscanthus, and poplar. It's important to note that the poplar system actually had a very healthy understory, so we actually counted it as a polyculture. And then we had our native grass mix that had 5 different species, our early successional system that had anywhere between 11 and 13 different species, and then the restored prairie system typically had about 18 different species. So the question they wanted to ask here was, how do these different cropping systems contribute to carbon accumulation? And so I've had kind of several discussions on soil carbon pools today, and thinking about the theoretical framework in which how we think carbon cycles in our agricultural systems. But just for simplicity's sake, in this study we thought about carbon as being in 3 distinct pools. So typically in agricultural systems, we typically just focus on one value, and that's total soil carbon. But any soil scientist will tell you that this number takes years to be able to detect differences, in large part because it is, it's the majority of the total carbon is in this passive pool. So when we want to try to detect differences in carbon accumulation across different treatments, it becomes really important to try to separate out different pools and understand how carbon might be accumulating at different rates. So an easy, distinct way to do this is to think about carbon in the labile or active pool, where carbon persists in the soil from anywhere from a day to up to a year. You have an intermediate pool where carbon persists from a few years to up to a decade, and then you have this very large passive pool of carbon that's really inaccessible to microbial activity and persists in the soil for hundreds to thousands of years. And so the research question here was, does perenniality lead to greater active, slow, and passive soil carbon pools compared to annuals? And are soil carbon pools greater under more diverse cropping systems? And again, we're very interested in this monoculture versus polyculture comparison. So in terms of some field methodology, we had 2 sites, one at the Keller Biological Station at Michigan State and one at the Arlington Research Station that's associated with the University of Wisconsin. And again, we're able to look at the 10 different systems across that annual perenniality gradient as well as the diversity gradient, where we had a randomized complete block design. We soil sampled in the fall of 2013, so when these experiments were in their 5th year, so the perennials are very much established at this point. And we took 1 meter deep cores and separated them into 4 different intervals. And so in terms of determining the soil carbon pools, there's so many different ways that you can do this, but for this experiment we decided to use respiration and long-term incubations, as well as chemical analysis using acid hydrolysis to be able to separate out those pools. So what we did was, the first thing after we collected our soil samples was we determined total carbon using an elemental analyzer. Then I established a 322-day-long incubation where I tracked CO2 very frequently early on, and then as time went on, it was every other week to once a month. And then at the end of the experiment, I used acid hydrolysis, which basically means I boiled soil in acid at very high temperatures to release the remaining biologically available carbon, and then determine total carbon after the acid hydrolysis treatment. And so how do I actually assign values to these different pools using these methods? So here you can see the respiration rates early on were quite high. This is where the carbon is very accessible to the microbial community. So, that reflects the labile carbon or active carbon pool. Then over time, you start to see a reduction in respiration. Carbon is becoming less available to microbial communities. And you move into this slow and passive pool. But even though we have this nice trend over time with the CO2 production, we still didn't have actual values for the pools. So in order to get that, I used the 3-pool model and combined it with nonlinear regression, and basically was able to determine total carbon from the initial sampling, and then total carbon after the acid hydrolysis was our passive pool. And then using the nonlinear regression, tracking CO2 over time, I was able to estimate that active carbon pool, and then by process of elimination, calculate that slow pool. So what did we find? In terms of the active carbon pool, this actually was about 4% of the total carbon. This was at the Arlington station, so a high clay mollisol site. In green, we have our annual system. In blue, we have the perennial monocultures. In red, we have our more diverse perennial systems. And actually, we don't really see that much of a trend. We see that the restored prairie system is significantly greater than corn, but in between, we actually don't see any real distinct trends in the active carbon pool. At KBS, we see a different story where it's an alpha sol site, it's sandier. And we actually see that the more diverse perennial systems had about 2.5 times more active carbon than the annual cropping systems. But what was interesting is that the monoculture perennial systems were more on par with the annuals versus the perennials. And it got me thinking back to my first chapter where we weren't picking up differences in the more labile carbon pool between annual wheat and perennial kerns. And I thought, maybe perenniality isn't the only thing that matters. Maybe diversity is something that really has to be considered in terms of seeing rapid carbon accumulation. But then in the slow pool, unsurprisingly, we actually see the trends dissipate. And that is because we did this after 5 years of sampling, and we could expect that if we go back in 10 years, we might begin to see differences. I actually have soils from their latest deep core sampling, so might have a student do a long-term incubation to see if we see the same trends. But there are some important things to point out here. So even in the slow pool, actually the poplars start to jump out in both places, which is interesting in terms of thinking about how their root systems, which are much more developed compared to the other systems, could be contributing to this slow carbon pool. And then in terms of the passive carbon pool, we did not see any differences across the treatments, but we did see some interesting differences via depth and across the different sites. It was clear that the high clay mollisols have a much larger passive pool of carbon compared to the sandier alpha sols. So just to summarize this section, it was very clear that diverse systems accumulated active carbon more quickly than the monocultures, especially amongst perennials, but only at the alpha sol site. And perenniality alone was not enough to increase carbon accumulation. So just some important implications from this. Diverse perennial crops grown on less fertile soils could lead to significant and rapid increases in soil carbon accumulation. So, the first two examples that I've kind of laid out for you have been on-station experiments. We've learned so much from different long-term trials in terms of how different treatments can contribute to ecosystem services. But the work that I'm doing now is asking, can similar trends be found on actual farmer fields? And one of the really important things that we have to think about is, farmers have a lot of things that they're working on, and a lot of you are very well aware of this. They're trying to increase yields, they're trying to improve their soil health, they're trying to manage weeds, and there are some interesting trade-offs that can occur when they're trying to manage all of these tasks. And especially in terms of soil health, it's important to think of soil health and how they relate to different ecosystem services. So in this experiment, we focused on kind of 3 biological soil health indicators. Respiration is a very useful tool in terms of predicting how much carbon is actually in the system, and it's also very representative of microbial activity, but it also is very reflective of carbon mineralization, whereas the active carbon pool, also known as permanganate oxidizable carbon, has been shown to be a soil health indicator that reflects more stabilized processes and could be linked to carbon sequestration. Then we have soil protein, which is able to quantify the available pool of nitrogen. And we're actually still trying to determine if it's reflecting a source of nitrogen or a sink of nitrogen. But some research questions for this section have been, what management practices most influence soil health in corn production across Indiana, Michigan, Ohio, and Pennsylvania? And again, kind of getting back to this perenniality or diversity, which has a greater influence on soil health. So the methodology that we used for this experiment is a little bit different. In my department, the 50% of the faculty are actually social scientists, so it presents a lot of opportunity to collaborate with using cross-disciplinary approaches. So we worked with social scientists that developed a very large survey that was sent to every organically certified corn grower in the 4 states mentioned here. And if the farmers filled out the survey, they were offered a free soil health test. And we actually got a lot of interest. We just sent this postcard, and all they had to do was return it and saying they were interested in participating in our study. And so once we received that postcard, we sent farmers a packet of information explaining exactly how to measure their fields. We asked them to select a soil sampling area that was less than 5 acres and asked them to sample 10 cores or slices to a depth of 20 centimeters. And then we asked them to composite those soil samples and send it in along with an additional management survey so we could get some their cropping history. Once we received the soils were sieved to 8 millimeters and then ground to less than 2 millimeters. And then we analyzed the soils for chemical, physical, and biological properties. And then farmers received a comprehensive soil health test if they sent in the management survey, which the majority of them did, which was great. And just to give you an example of some of the questions that we asked the farmers on this management survey, the first was definitely wanting to understand their crop rotation. So we asked for 4 years of cropping system history, and an example of a farmer who grew organic certified corn, but also had 3 years of hay. And then we were also interested in quantifying the different types of tillage as well as the number of passes on which type of tillage that they were using. And in terms of county representation, across the 4 states, we had 73 counties represented, and in total, we had 197 soil samples where they actually sent in the management survey. So just some quick kind of farm characteristics from this survey. It was clear that most farmers had 2 to 3 crops in their rotation, so it was mainly corn and soybean, or corn, soy, and some wheat, or corn, soy, and alfalfa was also common. And then in terms of manure, most of them added manure to their fields. Majority actually did not incorporate cover crops. And then what was surprising to us was that such a large number of these farmers actually had some type of perennial present in the rotation. So how do different management practices influence soil health in corn production? One of the things that was really shocking to me when I first started exploring this data was the fact that we actually saw a decrease in soil health when we saw an increase in crop diversity. And especially in terms of the permanganate oxidizable carbon, so the active carbon test, and the mineralizable carbon. And this was something that I just was really shocked because, in the literature we continuously see that increases in crop diversity tend to benefit soil health. But what we also saw was a significant correlation with an increase in tillage frequency and crop diversity. So what this means is that farmers are tilling more to get more crops into their rotation. And this is, in some cases, having a negative effect on soil health. But we still wanted to dig a little bit deeper in terms of asking, what drives soil health on organic farms in the Great Lakes region. So I used subset regression to ask which variables were most important in terms of predicting different soil health indicators. So in terms of respiration, tillage and perenniality were the top ranked predictors. And again, you can see that mineralizable carbon definitely decreases with an increase in tillage frequency, which is, what we would expect. And then in terms of perenniality, we do see a slight trend where there's, greater mineralizable carbon under perenniality, under farms that had a perennial in their rotation versus not. But I still thought, this isn't really telling a full story. What else could we try to pull to understand more about this kind of perenniality factor? So then I started to think about the length of time that a perennial is actually present in the rotation. We actually see this really nice trend where if there's, a perennial is present for 4 years, that substantially has a greater mineralizable carbon compared to if there's, never a perennial in the rotation, 1, 2, or 3. So this again shows that time is a really important factor, and the more perennials that you have in your rotation, the greater potential soil health that you can have. So this work is clearly still in the works. I haven't had a chance to look at some of the other soil health indicators yet, but I think it's an important trend in terms of understanding the importance of perenniality, especially in these organic corn production systems. So just to summarize, organic farmers in the Great Lakes region utilize a wide range of management practices to maintain crop rotational diversity. However, we actually see that crop rotational diversity has a complex relationship with soil health, and this is likely due to tillage frequency. We see that an increase in crop rotational diversity leads to an increase in tillage, which looks like it decreases soil health. However, when we see an increase in perennials in the rotation, we see a decrease in tillage and an increase in soil health. And I think this really just reflects the importance of conducting on-farm research and trying to see how different management practices that we focus so much on station trials, do we see the same trends in actual farmer fields. And that is actually something that we're actually seeing quite different results. So just some overall conclusions. Perennial grain crops have the ability to reduce nitrate leaching and increase nitrogen use efficiency relative to annual crops. Diverse perennials were able to accumulate 2.5 times more carbon relative to annuals and monoculture perennials in the active carbon pool. We also see significant trade-offs associated with crop diversity in organic corn production. And perenniality appears to increase soil health more so than crop diversity in organic crop production on farmer fields across the Great Lakes region. So with that, I'd just like to acknowledge technicians in my lab. Also collaborated a lot with Steve Coleman at OSU. Also, I'd like to acknowledge Phil Robertson, who's my PhD advisor at Michigan State, and also worked closely with people in the SNAPS lab. And then Doug Jackson-Smith is the social scientist that we collaborate a lot with at OSU. And then just like to acknowledge my funding sources and be happy to take any of your questions. Yes.
Since you showed your root systems and your perennial systems going sometimes more than 2 meters deep, do you think it would be of value to look at some of your carbon pools in that greater than 1 meter deep depth range?
Yeah, that's a great question. So we only sampled down to 1 centimeter or 1 meter because it's a lot of work to get deeper. But what was surprising from the annual and perennial Kernza experiment was we actually didn't see significant differences at lower depths, which was really shocking to us. We expected to see greater roots throughout the entire profile. So not sure that it would help to go deeper for the perennial Kernza research. Now, what I didn't show was root data from the biofuel cropping system experiment. And there we actually only looked at fine root production in the top 15 centimeters. And we actually saw significantly greater fine root production in those diverse perennial systems, which likely explain the greater active carbon accumulation. Yes?
What does it mean, tillage frequency of them?
Oh, could you repeat that, please?
Yeah, in one of your last slides, you had the tillage frequency in the x-axis.
I'm still not understanding.
Tillage?
Oh, tillage frequency. Yes. So great question. So in the survey, we asked for them to quantify the number of passes per year. And so we just totaled that value. So it's the sum of the number of tillage passes that they use throughout their rotation.
So is it reasonable for someone to have 30 passes per year?
Well, if they are trying to get in, multiple cover crops, some of them had vegetable crops that they were growing, so tilling in between each of those at every, before and after planting. That's what they reported. And when I showed this data to a group of conventional farmers, they said, yeah, of course, they till all the time. You're going to see a different trend with conventional farmers because a lot of them are able to be no-till. So it's an interesting trade-off in terms of organic versus conventional farming. Yes?
Did you look at, so in that study with the organic corn, so you showed the one that had 1 year of corn and 3 years of hay. Did you make any assessment about potential biomass that was incorporated with some of these extended rotations?
We weren't able to do that. It would be really great to go back to some of these fields and do intensive sampling. So we really relied on farmers to send soils to us. So we don't have any above ground biomass or below ground biomass data to correlate with this, but we have talked about selecting a core group of these farms across the 4 states and doing a more in-depth sampling, which would be really fun. Yes?
Christine, so it seems to answer the question you started out with, it's neither, it's tillage, right? Is the answer to the question, is that right?
Yeah, if you can reduce soil disturbance, that's the most important thing, and you can get that through perenniality. So I would kind of argue that perenniality is the answer. Yes?
I was wondering if you also looked at the nutrient composition of the root as a function of rooting depth. Was there any difference?
Yeah, so that's a really great question. We looked at carbon and nitrogen ratios all throughout the soil profile, and we saw that the perennial system actually had higher carbon to nitrogen ratios compared to the annual wheat. So again, they're able to retain a lot of carbon in their root systems, and that could be why they aren't decomposing as quickly with lower nitrogen, which could be why we didn't detect any differences in the active carbon flow. Yeah, it's a good question. Yes?
Based on your data, do you think that the permanganate oxidizing the carbon is a good index of soil health?
That's another really great question. I tend to think that respiration's a better measurement because it's more sensitive. I think that it's important to measure both because they're showing different processes in terms of how carbon is being cycled. So I think they both have value, but I do think from a farmer standpoint and being able to, compare two fields and really see drastic differences, respiration is the way to go.
Is your permanganate oxidizing the carbon correlated with any other indices of soil health information?
It's more correlated with total carbon, which would explain why we don't see as much of a difference. Yes?
So following up on Mike's question.
Yeah.
Respiration, there are a number of respiration tests out there in the soil health sphere, and when you say that, what kind of respiration tests do you need?
Yeah, so we just use a 24-hour respiration, and work out of, Steve Coleman's lab has compared a couple different lengths of incubations and different sieving techniques, and yes, there are so many different ways, and we're still kind of figuring out which is the most sensitive and accurate. We know that respiration is, the good thing about it is it's so sensitive and can detect differences, but it's also really variable. So it's something to really consider and be cautious about. Any other questions?
Have you thought about why your diverse perennial systems may have accumulated more soil carbon
Yeah.
than perennial systems at one point?
Yeah, so what I didn't show was fine root production from that cropping system experiment. We actually saw greater fine root production in the more diverse perennials, which is likely the result.
So why? Why more fine roots?
Yeah, so fine roots turn over more quickly.
No, I understand what they're doing, but I'm understanding I want to know where they're coming from.
So I think that the more diverse systems are just allocating more fine roots versus coarse roots. So we also looked at coarse roots by depth, and switchgrass and miscanthus had greater coarse roots compared to the diverse perennials. So it's just a shift in allocation.
So what if you took perennial from the diverse grouping that produced fine roots and grew it as a monoculture? Would you see more soil accumulation over that?
Well, switchgrass was present in each of those diverse systems, and then the switchgrass in the monoculture, we saw less fine root production. So what we think is happening in these more diverse systems is some type of plant complementarity, where perhaps the legume in that restored prairie is leading to greater foraging of fine roots. And so there's this, just different rooting mechanisms in monoculture versus polyculture systems that I think is contributing to that.
So it's not diversity per se, because you could put 3 tall grass species in there and it wouldn't be any different than growing one in monoculture, would it?
Well, if we, yeah, I mean, we know that plant competition happens in intra and interspecific systems, but we didn't see any increase in vine reproduction within the switchgrass system where we see multiple, there's plant competition happening, but we just didn't see that effect. So one way that we could test this would have an experiment like Cedar Creek, where they have monoculture systems and diverse systems, and have each species present individually and as part of a diverse group. Some would be able to tease apart some of those different effects. Any other questions? Thank you.
Thank you. That was a great presentation.
Thank you.